Welcome back to Things I Know Nothing About. I'm Maya, and as always, I'm joined by Alex, who's about to make me feel both amazed and slightly terrified about the state of modern science.
Great to be here. And today's topic is genuinely one of the most exciting things happening in medicine right now.
Okay, so hit me. What are we talking about?
Cancer, specifically, the drugs that scientists said could never exist and that now do. Dana-Farber Cancer Institute just published their annual 10 breakthroughs giving us hope list for 2026, and two items jumped off the page. First, menin inhibitors just got FDA approved for roughly 40% of acute myeloid leukemia cases. Second, RAS inhibitors, targeting what was literally called undruggable for over 30 years, are now in phase 3 trials for pancreatic cancer.
Pancreatic cancer. That's the one everyone dreads, right? The survival rates are terrible.
Devastating. 5-year survival sits around 13%, and for metastatic cases, it drops below 5%. It's projected to become the second leading cause of cancer death in the US by 2040. There just haven't been good targeted treatment options until now.
So let's start at the beginning. What is RAS, and why did everyone think you couldn't drug it?
RAS is a family of proteins, KRAS, NRAS, HRAS, and they act like molecular switches inside your cells. When a growth signal arrives, RAS flips to the on position, tells the cell to divide, and then flips back off. Clean. Controlled.
Sounds like a light switch.
Exactly. Except when KRAS gets mutated, the switch gets stuck in the on position permanently. The cell just keeps dividing. And KRAS mutations show up in about 30% of all human cancers, lung, colon, and especially pancreatic, where roughly 90% of tumors carry a KRAS mutation.
90%. So almost every pancreatic cancer patient has this broken switch.
Virtually all of them, scientists discovered the KRAS oncogene back in the early 1980s, over 40 years ago, and almost immediately they wanted to build a drug that could block it. The problem, KRAS is tiny. It's this smooth little protein with no obvious pockets or grooves where a small molecule drug could grab on.
Like trying to stick a post-it note on a marble.
That's a perfect analogy. Researchers spent decades trying and failing. By the late 90s and early 2000s, the scientific consensus was pretty brutal. KRAS is undruggable. Move on. Focus on something else.
But clearly, someone didn't get the memo.
Right. In 2013, a researcher named Kevan Shokat at UCSF discovered something nobody expected. One specific KRAS mutation, called G12C, actually creates a tiny hidden pocket in the protein right under what's called the switch II region. It only appears when the protein is in its inactive state, but it's there.
A secret door in the marble.
Beautiful way to put it. Shokat's lab showed you could design a small molecule to slip into that pocket and essentially lock KRAS in the off position permanently. That discovery blew the field wide open. By 2021, the FDA-approved sotorasib, brand name Lumakras, made by Amgen as the first ever direct KRAS inhibitor. It targeted G12C mutated non-small cell lung cancer.
Okay, so historic moment, but that was for lung cancer, not pancreatic?
Correct? And here's the catch. The G12C mutation is common in lung cancer, but the most common KRAS mutation in pancreatic cancer is a different one. G12D, around a third of pancreatic cancers carry it specifically, and G12D is even harder to target than G12C because it doesn't have that same accessible pocket.
So if G12C was the secret door, G12D is the vault.
The vault with no keyhole, at least that's what people thought. But several groups have now found ways in. One early breakthrough was MRTX 1.33, a non-covalent inhibitor that can bind both the active and inactive forms of the KRAS G12D protein. It was the first proof of concept that you could directly target G12D with a small molecule.
When did that happen?
Preclinical data came around 2021, and it moved into phase I clinical trials. Then you've got HRS 4642, another G12D inhibitor with early phase I data showing tumor shrinkage in lung cancer patients. Veristem Oncology reported preliminary results from their VS7375 program in late 2025. That one's designed to block KRAS G12D in both its active on and inactive off conformation.
Dual mechanism, like locking the switch from both sides.
Precisely. And the big news from Dana-Farber's 2026 list is that a novel RAS inhibitor for pancreatic cancer has moved into a phase 3 clinical trial, led by Brian Wolpin, who directs the Hale Family Center for Pancreatic Cancer Research there.
Phase 3, that means it's being compared head-to-head against standard treatments in a large group of patients.
Exactly.
Phase 3 is the last major hurdle before seeking regulatory approval. Early results are reportedly promising. And there's an incredible quote from Andrew Aguirre, co-director of Dana-Farber's Center for RAS Therapeutics. He called the revolution in targeting RAS, and I quote, one of the biggest therapeutic advances in the history of clinical care for pancreatic cancer patients.
That's not a small statement for a disease where the standard of care has barely budged in decades.
Not at all. When researchers use the phrase history of clinical care for pancreatic cancer, they mean it. This is a disease where gemcitabine, a chemotherapy drug approved in the 90s, remained the backbone of treatment for over 20 years. Progress has been agonizingly slow. RAS inhibitors represent the first time we can directly go after the primary genetic driver.
All right, let's switch gears to the other big breakthrough. Menin inhibitors. Tell me about those.
So, this story starts with acute myeloid leukemia, or AML. It's one of the most aggressive blood cancers. It moves fast, it's hard to treat, and outcomes for relapsed patients are grim.
How does menin fit into the picture?
Menin is a protein in the nucleus of your cells. Normally, it helps regulate which genes get turned on and off during blood cell development. It works closely with another protein complex involving KMT2A, also known as MLL. Under normal circumstances, this partnership is tightly controlled.
Let me guess. Something goes wrong with the partnership.
Two things, actually, in some AML patients. The KMT2A gene gets rearranged, essentially broken, and fused to another gene. This creates a mutant protein that hijacks menin to permanently activate genes that drive leukemia. In another subset, a gene called NPM1 gets mutated, and it similarly co-opts the menin-MLL machinery.
So menin isn't the villain itself, it's being used by the villain?
That's a great way to frame it. Menin is getting weaponized. Scott Armstrong at Dana-Farber spent over 20 years studying this biology. His lab figured out exactly how the mutant MLL protein and menin conspired to activate the leukemia program. Then they set out to find a drug that could pry them apart.
Break up the partnership.
Literally. In 2019, working with Syndax Pharmaceuticals, Armstrong's group described a class of menin inhibitors that could eradicate leukemia in preclinical models. The compound that emerged from that work is called revumenib, brand name Revuforj. It's an oral pill taken by mouth.
And it just got approved.
2 approvals, actually. November 2024, the FDA-approved revumenib for relapsed or refractory acute leukemia with KMT2A translocations in both adults and children. Then in October 2025, a second approval came through for AML with NPM1 mutations, making it the first and only menin inhibitor approved for multiple subtypes of acute leukemia.
Both adults and kids. That's significant for a cancer drug, isn't it?
Very much so. Pediatric approvals are less common, and KMT2A rearranged leukemia disproportionately affects young patients. Having an option beyond intensive chemotherapy is meaningful.
Jacqueline Garcia at Dana-Farber called it a monumental step forward. What's next?
Combination therapy. Revumenib works, but researchers want to see if pairing it with other treatments, chemotherapy, other targeted agents, can produce even deeper, more durable remissions. Those combination trials are already underway.
And these two subtypes together cover about 40% of all AML.
Approximately, yes. KMT2A rearrangements and NPM1 mutations combined account for roughly 40% of AML cases in both children and adults. That's a huge fraction of patients who now have a fundamentally new treatment option that didn't exist 2 years ago.
Zooming out, we've got menin inhibitors for leukemia, RAS inhibitors for pancreatic and other solid cancers. Both of these were supposedly impossible targets. Is there a pattern here?
There really is, and it goes beyond just these 2 stories. Dana-Farber's 2026 list also highlights protein degraders, drugs that don't just block a problematic protein, but tag it for destruction by the cell's own recycling machinery. Eric Fisher at Dana-Farber said he thinks this coming decade could be the clinical decade for protein degradation.
Instead of blocking the lock, you shred the whole door.
Love that metaphor. Then there are personalized cancer vaccines designed to train a patient's immune system against their unique tumor. Liquid biopsies that can detect cancer relapse from a blood draw, radioligand therapies delivering targeted radiation directly to tumors.
It feels like the toolkit just exploded in the last few years.
That's exactly what happened. For decades, cancer treatment was surgery, radiation, and chemotherapy, a blunt toolkit. Then immunotherapy arrived and changed everything for some cancers. Now we're entering this era of precision where we understand the molecular wiring of individual cancers well enough to design drugs that go after specific broken components.
And the undruggable label?
Increasingly obsolete, the lesson from KRAS is that undruggable often just meant we haven't figured it out yet. Shokat found the hidden pocket in 2013. Armstrong cracked the menin-MLL interaction. New technologies, cryo-electron microscopy, AI-driven drug design, better understanding of protein dynamics, keep revealing vulnerabilities that were invisible before.
So when Andrew Aguirre says this is the biggest therapeutic advance in the history of pancreatic cancer care, he's not being hyperbolic.
Not at all. For a disease that's had a 13% 5-year survival rate and where the standard of care barely changed for 2 decades, directly targeting the primary genetic driver isn't just an advance, it's a paradigm shift. We're not there yet. These are still clinical trials and not every patient will respond, but the trajectory is genuinely historic.
All right, let me try to sum this up. RAS, specifically KRAS, is mutated in 30% of all cancers and almost all pancreatic cancers. Scientists spent 40 years calling it undruggable. Then Kevan Shokat found a hidden pocket in 2013. Sotorasib got approved in 2021 for lung cancer, and now we're in phase 3 trials targeting the hardest KRAS mutations in pancreatic cancer.
Perfect summary.
Meanwhile, menin inhibitors like revumenib went from lab discovery to 2 FDA approvals in about 5 years, covering 40% of AML patients. And the broader theme is that the concept of undruggable is crumbling across oncology.
Couldn't have said it better. The next few years are going to be remarkable.
Cancer's worst year indeed. Thanks for listening, everyone. We'll see you next episode.
Until then, stay curious.